The first time you learned about osmosis, you just learned the terms like hypertonic, hypotonic, and isotonic. If you remember what those mean, a hypertonic solution is one that has more solute and less water available. So imagine taking a whole bunch of salt or sugar and mixing it in water. That's a hypertonic solution. If we put a red blood cell or we put some sort of animal cell into that, water is going to move from where there's more water available, which is inside the cell, to the hypertonic solution outside the cell, and that cell is going to shrivel up. A hypotonic solution is where there's less solute and there's more water available. So if we just take a beaker of water with very minimal, like think maybe a quarter of a teaspoon of salt or sugar mixed into it, and you put a red blood cell or any other type of animal cell into that, water is going to enter that cell. It's going to move from where there's more water available to where there's less water available, so into the cell. Cell will swell and eventually possibly burst. Animal cells prefer isotonic solutions, where the outside of the cell solution is exactly the same as the inside of the cell solution. So water enters and exits the cell in the same rate, and there's no net water movement. So it's entering and exiting at the same rate. Plant cells, on the other hand, prefer a slightly hypotonic solution on the outside because they prefer that water comes into the cell. That fills the plant cell with water and pushes the membrane against that cell wall. And that's what we call turgor pressure. That makes a plant firm and it makes it stand upright. That's kind of its structure. It comes from that. Now, the second time that you learned about osmosis, you learned about water potential. Right? So water potential is a calculation that can help us predict which way water is going to move. And our overall equation for water potential is overall water potential is equal to the pressure potential plus the solute potential. So let me just draw a little line here. In most problems that we saw, the pressure potential was 0 because the cell or the beaker was just sitting in an open container. So there was no pressure pushing on the cell or the water, and there was no pressure, like think of a straw, like sucking up water. There's no pulling pressure either. So in most of the problems, and if you would see this on the AP exam, most likely the pressure potential is going to be 0. If we have a beaker of pure distilled water where there's no solute mixed in the water, our solute potential is also going to be 0. But as we add solute, that lowers and makes this number negative. And let's refresh our memory why. So in an open beaker with no pressure of distilled water, all of these water molecules are available to go somewhere, right? So if we put this somewhere else in, or we put a cell inside of this, the water has the potential to move into the cell. But now if I look at this beaker where I've added a little bit of solute, maybe that's some sugar that I've mixed in, water molecules, because they are polar, are attracted to other polar solutes. That's how we dissolve things, if you remember that. So these sugar molecules are dissolved because they're surrounded by water molecules. These water molecules that are surrounding the sugar are busy, they're occupied. They do not have the potential to move anywhere. So that lowers the the solute potential because those water molecules are not available, right? They have less potential to go somewhere. Now, if I add even more solute to a solution, more water molecules are going to be occupied and busy, and they're going to be less available water molecules to move. So that lowers the solute potential even more. Now, looking at a few problems, I kind of use these same numbers here for this first problem here. If a solution has an osmotic potential of -5 bars, so think water potential if you would see this, and another has an osmotic potential of -8 bars, which one has the greater molarity? Now, molarity is a measurement of how much solute there is. So which one has more solute? This one. All right, so that's going to be our -8 bars would be the answer for number 1. If we compare those two solutions, which one is, this should say more hypertonic to the other, which one has more solute and less water available, that's going to, again, be the -8 bars. 3, which one has the greater water potential? Now, be careful with this. We're dealing with negative numbers. We are comparing -5 to -8. Which one is the higher number? It should be -5, right? And going back to the science, this solution has a greater potential to go somewhere because there's more water molecules available. So that might be a helpful way to think about when you're talking about greater versus less water potential. Which one has more potential to move somewhere? Now, going back to our formula, those first 3 questions dealt with kind of where I drew my line, just looking at the overall water potential of a solution. Now, sometimes they don't give you this number like they did in numbers 1, 2, and 3. Instead, they give you some clues, like they give you the concentration of the solution, like they would say it's 2 molar or it's 0.5 molar. They could give you the temperature of the solution, and they ask you to calculate solute potential. So really, they're just giving you clues to help you figure out that number. So let's look at number 4, A and B, to help us refresh our memory on how to use that equation. So number 4 says, calculate the solute potential of a 0.1 molar sodium chloride solution at 25 degrees Celsius. Again, they're not giving you the solute potential, they're giving you some clues or some hints. Now I'm going to flip to the next slide because they already have it worked out for you. So if we look up here at the top, we have our calculation to figure out solute potential, which is -I CRT. -I is the ionization constant. If they're talking about a sugar being dissolved in the solution, that ionization constant is 1 because sugars don't break apart in water, they don't ionize. But if they tell you that it's a salt, like sodium chloride, when we mix that in water, it breaks into 2 ions. It breaks into a sodium ion and a chlorine ion. Therefore, for this solution, the ionization constant is 2. So our number goes there. The concentration they tell you is 0.1 molar. So that's going to go for our C. The R is a constant that is always this value. You do not have to memorize that. That is right on your formula sheet for you. And then our temperature, we convert Celsius to Kelvin by taking 25 degrees Celsius and adding 273 to get Kelvin. So if we multiply all of this together, our solute potential for this solution is -4.95. Now, just for the heck of it, let's use our overall equation where water potential is equal to the pressure potential plus the solute potential. Nowhere in this problem do they tell you anything about the pressure. They don't say that there's a pressure being pushed or a pressure being pulled on the solution. So we can assume that this is an open beaker, an open container, and our pressure potential is going to be 0. Therefore, when we add in our solute potential, which is -4.95, the overall water potential for this solution is going to be -4.95. Now, how do we use water potential? That's where A comes in. Now, let's say that we find a cell that is 0.15 molar, and we take that plant cell and put it into this beaker, right, with this water potential. What's going to happen to that cell? That's where water potential comes in. So now we repeat this equation, but if you notice, the only thing that's different is we have a different concentration. So the beaker was 0.1 molar and the inside of the cell is 0.15 molar. So we repeat that and figure out that the water potential of the inside of the plant is 0.743. So if we compare those numbers, out here in the beaker is negative 4.95. And if this is my plant cell, the water potential is negative 7.43. Again, what do those numbers mean? The lower the number, the more solute there is, so the higher the molarity. The higher the number, the less negative, the less solute there is. So if we compare this, the inside of the cell, or I should say the outside of the cell, is hypotonic to the inside of the cell. So what's going to happen? Water is going to move into the plant cell, increase its mass, increase the pressure in there, and it's going to swell. So this is how we use water potential to help us calculate and answer osmosis problems. If you would like more water potential problems, if you just do a quick Google search, they are everywhere with answers. Or if you would like ones from me, just let me know. I will be happy to email them to you. The last part of this video looks at some active transport processes such as endocytosis, exocytosis, ion channels that use ATP, and so on. Here is an FRQ from the 2018 question or exam, and it's talking about cystic fibrosis. So there's a protein called the CFTR protein that's a gated ion channel that requires ATP in the cell membrane. It allows chloride ions to diffuse across the membrane. Take a second, look at the prompt, and then we'll work through this prompt together. So let's think about this. So we have our cell membrane. I'm just going to draw like a curve for the membrane. And then we have a protein in the membrane that is used in uses ATP to transport chloride. Okay, so this is an active transport thing. Now, the first thing is talking about where does this protein come from? Obviously, the cell is going to be making this protein. Think about the processes involved in making a protein. Okay, so looking at the answer key on the next slide, here's our endomembrane system, right? So the nucleus has the DNA that codes for the RNA. The RNA is going to be sent to the ribosomes on the rough ER. The rough ER will send the made protein to the Golgi, which is going to package it. And then that's going to be sent to the vesicle to make that membrane protein, right? So let me do that again. So now we have our membrane and we have our protein. That's where it came from. It came from protein synthesis. Part B now is going to bring in some active transport questions. So if this is not working, so if cystic fibrosis comes about because we don't have this protein, it's not functioning, what's going to happen to the chloride concentration in the cell? And it's kind of saying, well, if we can't move chloride ions, this is going to stop chloride movement and it's going to get stuck on one side, making one side more hypertonic. Okay, so for example, if this chloride ion normally moves chlorine out, and it's going to move it out, and it's going to move it out, but it's not working, where is the chloride going to build up? It's going to build up in here. That's going to make this side of the cell more hypertonic. So what's going to happen? We're going to see water moving into the cell. That's an issue. Okay. That would be a question where you're looking at just kind of a process, and we're tying two different processes together. We're talking about protein synthesis, and we're talking about active transport and then osmosis as well. So this is a good question because it asks a lot of different concepts in a very simple question. The last thing, I like that at each of the end of these videos, they give you stuff from the AP curriculum. This is a nice little checklist for you to say like, ooh, yes, I got this, or no, I don't. And those are some areas that you can focus on reviewing for yourselves. Thanks again, guys.